Welcome to Chapter 8, Microbial Genetics, Part 1. In this chapter, we're going to be fulfilling objective number 2, which is to provide the students with the knowledge necessary to comprehend the fundamentals of microbiology, especially microbial genetics. In Part 1, we're going to cover structure and function. In Part 2, we'll talk about mutation, genetic transfer and recombination, genes and evolution. Under the category of structure and function, we're going to review the structure of nucleic acids, since that's what we're going to be talking about. We're going to talk about the difference between genotype and phenotype, especially how it applies in microbiology, DNA and chromosomes, DNA replication, and RNA and protein synthesis. First, let's review the structure of nucleic acids and their monomers, nucleotides. Let's start with nucleotides. Now you'll remember this diagram from Chapter 2. Oh no, Chapter 2! Yes, there's lots of chemistry in this Chapter 2. But you'll see down here we have our sugar, ribose. It's a 5-carbon sugar. Okay. And over here we have a phosphate group. Remember, it's the phosphate group that makes a nucleic acid an acid. And then over here we have our nitrogenous base. We usually call this just a base. Shorten it down. Now you hook these together, and if we are using this diagram, these are the sugars and the phosphates, sugar phosphates hooked together. Okay, that's the backbone of the nucleic acid. Then the bases turn inward. Then we can get another strand of DNA, and these bases are going to hold hands. And this is what creates the famous double helix. Now, here's something I want you to remember. A, adenine, always goes with thymine. A goes with T, G goes with C. This is always the case for DNA. So A is always going to match up with T, G is always going to match up with C. And this is part of how the cell can make exact duplicates of its DNA. We're going to talk about this more when we talk about DNA replication. Now let's talk about the difference between genotype and phenotype. Let's start with eukaryotic organisms, which you're more familiar with. In fact, if you've had Biology 201, we talked about Mendel's pea plants. That's why I have a picture of a pea flower here. So the genotype in eukaryotes are all the genes that are present in the cell. That is the genotype. The phenotype is what's expressed. Now in eukaryotes you have 2 copies of each gene, generally speaking. Fungi are different. But for most of them you have 2 copies. And so you have a dominant and you have a recessive gene, and the dominant one is the one that's expressed. Well, in bacteria and archaea, they only have 1 copy of each gene. So we use the terms genotype and phenotype a little bit differently. Genotype are all the genes, same as with eukaryotes. Phenotype are the genes that are turned on, the genes that are currently being expressed. So let's talk about an example. Let's talk about the life cycle of E. coli in places where you don't have water treatment. So E. coli are happily living in your intestines, and they're using their fimbriae to hold on to your intestinal cells. But those intestinal cells are constantly being shed, and E. coli leave in the feces. So eventually, those E. coli are going to make it into the local water supply. We're going to say that it gets washed out of the outhouse and down into the river. Now, E. coli don't particularly want to live in the river. They want to live in somebody's intestines. That's what they're built for. So they stop making fimbriae once they leave human intestines. They're floating along without fimbriae, just waiting to be drunk. And once they're drunk, they don't produce fimbriae in your mouth. They don't want to stay in your mouth. They don't want to stay in your stomach. They start producing fimbriae once they enter the small intestine. Then they can grab a hold again. Okay. I hope that hasn't grossed you out too much. But you see the difference between genotype and phenotype. The E. coli that are swimming around in the river have the genes for fimbriae, but they're not turned on. So the phenotype is to not have fimbriae. Once they get into the human intestines, those genes are turned on, and the phenotype is for fimbriae. Now let's talk about chromosomes. Okay, in eukaryotes, chromosomes are linear. Okay, the purple lines are the sugar phosphate backbone, the red lines are the bases holding hands, and in most eukaryotes, fungi excluded, you have two copies, one from the mother, one from the father. In bacteria and archaea, most of the time, the chromosomes are circular. You don't have hands. Now, this can be handy in certain circumstances, but we're not going to cover that in this class. But what you need to remember, most eukaryotes have linear chromosomes. Most prokaryotes have circular chromosomes. Now, from there, they get packaged, and we don't really need to worry about that in this class, but let's talk about making a copy of the DNA. We call this DNA replication. Anytime a cell divides, whether it's the cells in your dermal layer of your skin or the prokaryotic E. coli in your gut, every time those cells divide, each cell needs to have a copy of the DNA. Otherwise, they won't know what proteins to make, so you have to make copies. So, when we do this, we split the double helix, so we have a template strand, one that's going to be a copy for the new strand. And we make this new strand by hooking the sugars and the phosphates, phosphates, sugars, phosphate, sugars, phosphate, sugars together. And we always go from the 5' end to the 3' end. Now, if you want to know what that means, get with me during class, but what you need to remember, always goes 5', 3'. The other side, here's the 5' end, 3' end. So they are anti-parallel. So what happens is DNA polymerase comes along and matches up the bases that go with each other. And they come in as triphosphates. Then as they get hooked together in that's right, a dehydration reaction, we clip off 3 of these phosphates. That provides the energy for the reaction. So the DNA polymerase, which is the enzyme that does this, hooks them together. Then we have another one that comes in. So what's going to match with the C? That's right, we're going to have G matching with the C, and so on and so forth. Now let's expand out and see what's happening on the entire chromosome. Now, for me, it's easier to remember what's happening by remembering the enzymes that are doing the jobs. So, the parental DNA is unwound, okay, by a helicase. Helicase unwinds the helix, unzips them. Then you get proteins that stabilize the single strands so they don't go back and hold hands with each other. Now, DNA polymerase sets down and starts polymerizing the new strand like we talked about in the previous slide. But it can't get its mouth around a single strand. It can only get its mouth around a double strand. So, an RNA primer, we call it primase in this situation, it can get its mouth around a single strand. Doesn't care. So it makes a DNA-RNA hybrid. Then the DNA polymerase can grab a hold and start polymerizing. Now, on the strand that's going 3' to 5', it can go continuously. We call this the leading strand. On the other strand, the DNA polymerase keeps running in to previous primers. As this gets unwound, primase comes up, makes an RNA primer, DNA polymerase attaches here, goes down, runs into the primer, falls off. So what happens is, we call this the lagging strand. And these segments of DNA are called Okazaki fragments. Now, we can't have an RNA-DNA hybrid. It's not as stable as a DNA-DNA strand. Another DNA polymerase comes through and replaces the RNA primer and replaces it with DNA. Then DNA ligase comes through, hooks the pieces together, and we get one continuous strand. Now I'm going to show you an animation. And in the interest of making sure that copyrights are kept to, I got it from McGraw-Hill. And this is the website where you can go to. And here comes the animation.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides, complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
Okay, so we have a newly divided cell. It's got fresh new DNA, and now it wants to make proteins. That's what bacteria do. That's what cells do. So what we do is we undergo transcription. That's where we make an RNA copy of a DNA template. So in this diagram here, we get an RNA polymerase, and unlike DNA polymerase, it doesn't need helicase to unwind it. For one thing, it's only unwinding a specific portion of the DNA, just one gene. So it sits down, unwinds the DNA, breaks apart the bases, and it makes an RNA copy of the DNA template. Now, one thing you may have noticed, there's a U here. What's the U doing? RNA doesn't have a T, doesn't have thymine. It does have uracil. DNA doesn't have uracil. Uracil will hold hands with A. Okay, so when we go from DNA to RNA, no T's. Okay, so we make a messenger RNA, an mRNA, from the template. Then once that gets done, the mRNA falls off, and we're ready to make some proteins, which is called translation. Okay, to make the protein, we take the mRNA and we attach a ribosome to it. Now, ribosomes are made out of protein and RNA. The RNA is called rRNA, or ribosomal RNA. We also have transfer RNA. Transfer RNAs have amino acids on them, and they carry the amino acids from the cytosol into the ribosome. Okay, so the first thing that happens, and this is called initiation, is the mRNA for methionine, all proteins start with methionine, attaches to its matching 3 nucleotide word. We call it a codon. The part that matches it on a tRNA is called an anticodon. So they match up, A to U, A to U, C to G. Then the large subunit of the ribosome attaches to the small subunit of the ribosome, so that the part of the active site of the ribosome that's called the P site is over the tRNA. Now, ribosomes have three active sites. They have an E site, a P site, and an A site. You can think of the A site as being the addition site, the P site being the protein site, and the E site being the exit site. Okay. Another mRNA comes in that matches the next codon. Then what happens, coming down here, the methionine is moved from the first tRNA and is attached to the next amino acid that is on the other tRNA. Then the ribosome moves down one codon on the mRNA. mRNA stays straight, think of it as train tracks, and the ribosome as being a train, and it moves down. The empty tRNA leaves through the E site, goes off, gets another methionine. Then we have the next tRNA that matches the next codon comes in, and the lengthening polypeptide is moved over. Okay, and this keeps happening. Empty tRNAs go out through the E site, full new tRNAs come in through the A site. On the P site, the lengthening chain is moved over. This happens again and again and again. Okay, now when we get to the end, we have stop codons that do not code for an amino acid. There's no tRNAs that match the stop codon. When this happens, the ribosome subunits come apart, the last tRNA comes off of the mRNA, the protein, which has been folding into its active shape as it is made, gets taken off and it goes and does what it's supposed to do. The mRNA can be used to make more proteins. Okay, now I've got another animation for this because this is easier seen when all the parts are moving.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. There is also a third site called the exit or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of one codon. As the shift occurs, the two amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
Okay, the last thing for this part is we're going to practice making a protein. And we're going to start with the DNA, and I'm going to have you make an mRNA, and from there you're going to figure out what amino acids it codes for. So let's start out with the DNA. So you've got TAG, TGA, GGG, ATC. Now go back and rewind this if you need to write it down again. But write down from that DNA what RNA you're going to get. Now remember, T goes to A, A goes to U, G goes to C, C goes to G. Okay, the mRNA that you're going to come up with is AUG, ACU, CCC, UGA. Now, here comes the codon chart. So we look for the codon AUG. Now, I happen to know where it is. AUG codes for MET, also codes for start. So, MET is the next thing. Okay, that's the amino acid. Let's look for ACU. Oh, there it is right there. Codes for threonine. THR, that's all you need to remember. CCC, oh, let's look around. Oh, there's CCC, codes for pro or proline. And the last one, UGA, codes for stop. It's a very small protein. Okay, go ahead and practice as many times as you like, and we will be practicing in lecture. Now, if this has seemed like a lot, that's understandable. Go ahead and listen to this as many times as you like, watch the animations again, and we'll be going over this in class.